Good afternoon, and thank you to Nanopore for allowing me to come and talk about my research. So the aim of our group is really to understand the molecular mechanisms that drive kidney disease, and in particular, I'm interested in what drives diabetic kidney disease, for the purposes that diabetes is the leading cause of end-stage renal disease worldwide. And currently, the therapies that are used to treat the progression of this are lacking. And so there's a need to discover novel mechanisms to create new therapeutics to create a better thing for these patients. And so through a large body of work from our group and others, which I've referenced down here, using single-cell RNA sequencing short read, they found that there is a decrease in this factor called SRSF7 in the proximal tubule cells of kidneys. And these are serine arginine-rich splicing factors, which are shown here. It's a family of 12 different splicing factors. We're focused on SRSF7, and these are involved in alternative splicing. And basically, they aid in the recruitment and assembly of the spliceosome, which is the machinery necessary to perform splicing, where you splice out introns or exons. And you can see this tiny orange factor, which is SRSF2 in this picture, but SRSF7 performs in a similar manner at the beginning of this entire process to recruit these factors. And the point of alternative splicing is really to create transcriptional diversity. From one gene product, we can get many different isoforms that might have different functions. And so these are the main alternative splicing patterns that have been observed. They're not exhaustive, but this can lead to altered function due to splicing in or out of various functional domains, as well as altered expression due to either a non-coding transcript or something that is due to nonsense-mediated decay. So alternative splicing is also this additional regulatory mechanism outside of just expressing a gene. So there are various tools to assess RNA splicing. In the past, it was mainly using short-read RNA sequencing. However, as many of you probably know, this is lacking as far as how it can resolve individual isoforms because they have to be inferred. And so with the invent of long-read, we are now better able to resolve individual isoforms, usually the entire thing. And so this allows for more accurate transcript discovery and quantification. So to understand the function of this factor in kidney cells, I just want to give you guys a brief reminder about the kidney. It has an outer cortex and an inner medulla. And within the kidney, the functional unit is called the nephron. And we tend to focus on the proximal tubule cells, or the PCT cells, that are in the cortex. These are the most prone to injury and also highly metabolically active. And so we utilize this cell type. There is a immortalized human cell line of renal proximal tubule epithelial cells that we use to test this. And so we treated these cells with siRNA against SRSF7 and then submitted the samples for both short and long read sequencing. And I'll mainly focus on the long read results. And I just want to show an example of like what comes off the sequencer, which is just, this is showing the distribution of read lengths from this experiment up to like 4 kb. So seeing that we do indeed get long reads from this. And then when we look at after aligning and putting it into IGV, we can compare and see the differences between short and long reads. So at the top are the short reads, and they're just little snippets trying to figure out where they are. And then with our long reads, you can see this is the SRSF7 gene, which runs right to left. And many of the reads are, 1,800 to 3,500 base pairs, and they are covering the entire gene. So we're getting a better picture of what is actually happening at the isoform level. I also did an analysis to measure exon-exon junctions between the long and short reads for each sample. This was done using reg tools from this reference. And we found in every sample that long reads, even given the difference in coverage, were able to detect more exon-exon junctions, which is very critical for alternative splicing analysis, because that's what is typically used to understand the splicing that could be occurring. To understand the function of this, after we performed our RNA sequencing, we did gene set enrichment analysis and showed that in our knockdown cells that they had enrichment for inflammatory pathways. And then also noticed in our normal cells, they were enriched for like cell cycle pathways, which had us hypothesize that there might be some proliferation effect on these cells. So we did measure MKI67 expression and saw that this was indeed markedly reduced, and also did BRDU incorporation to look at the cell proliferation again and saw that this was also lower with SRSF7 knockdown. So not only does this factor, at least when it's present, seems to help preserve this, keep a anti-inflammatory environment as well as maintain proliferation, since when we don't have it, we have inflammation and a decrease in proliferation. Another analysis we did was using this package called Bamboo, which is specifically made for long reads and can quantify as well as do differential transcript usage analysis to understand the alternative splicing that is occurring. So we ran our RNA-seq data through this package, and it identified over 500 genes with differential transcript usage. And then we did gene ontology analysis to see which pathways these genes were involved in. And interestingly, again, we see these genes are involved in inflammation and cell cycle and also in splicing. So I just want to give one example of a gene we found that was in a cell cycle pathway called BANP. And interestingly, when we did the same analysis in our short read, we didn't find this gene. So this was only found with our long read sequencing data. And we use this package called isoform switch analyzer, which helps predict isoform switches, as well as any potential functional consequences of this. This is what the colors indicate. And so from just this one gene and this analysis, it predicted an increase in this 204 transcript and a decrease in the 228 transcript. And another thing I really want to point out is the gene expression between the two conditions is not different. So this is something you would miss if you just did gene level analysis. And then to validate this finding, I designed primers against basically the difference between the two transcripts was the inclusion or exclusion of this exon 5. So I designed primers to flank that exon and performed RT-PCR. And as you can see, we do in fact have a decrease in the 228 transcript and an increase in 204. And so as you can imagine, one way we can also test the function of this, since this is a cell cycle gene, is to target this individual transcript and see if it would have effects on cell proliferation. And this is something we want to do with not just this gene, but many others for future studies. The next question we wanted to know was, how does splicing change across mouse kidney epithelial cell types? And the perfect way to assess this is by using single-cell long-read sequencing. And so we use the 10x Genomics platform to do our single-cell, and then Nanopore has a part of this workflow. So we start with our cells. So for this experiment, I used 3 different mouse kidneys and did single-cell dissociation. We used the 3' GenX kit and targeted 10,000 cells. I will say that is a lot, and it does affect the coverage of your genes or transcripts per cell. So just keep that in mind if you're looking into your own experiments. And then you kind of follow the same 10x workflow until you get to the full-length cDNA generation. And because we can now sequence full-length cDNA, we just submit this for the sequencing, or the library prep and then sequencing. And for this experiment, we did 1 sample per flow cell. Again, this is probably not ideal with the amount of cells we targeted, but you can always sequence across more flow cells. And then finally, we used the Epitome Labs workflow for single cell to analyze this data. So I just want to start with some read and workflow statistics. This is just highlighting things like the median read length, which was over 800 for each sample, which is pretty typical for single cell RNA-seq. And then our median read quality was 22, which was much higher with this new implementation of Dorado. I had done some previously where it was much lower. So this is just continuing to improve. And then finally, from ONT's workflow, it will tell you how many high-quality barcodes it has identified or number of cells. And we can see for each sample it is around 10,000, so that's kind of on target. And then we get the expected number of genes and transcripts. And again, you'll notice median reads per cell is pretty low, but it is enough for us to detect the cell types that we expect, which I'll get into. So from this data set after QC, we identified over 16,000 cells from these 3 mouse kidneys across 20 different clusters. We then used our sort of in-house and published cell type annotations to annotate these cells. And we come up with this single cell UMAP showing very typical cell types that we would expect in the kidney, even some that are harder to detect with single cell, including podocytes and certain immune cells like T cells and B cells. So we were really pleased to see that, even given the lower coverage. Another thing we had noticed is that our very canonical cell type markers were consistently kind of split across 2 different cell clusters. And I'm not entirely sure why this is happening, but we are wondering if it is due to the long reads that we're getting now with this single cell workflow. And just to kind of show you what I mean, SLC5A2 is a very canonical PCT marker, and you can see it pops up not only in one, but another cluster, and same with SLC7A13 and SLC12A1. So as of right now, I'm treating them as different. I'm not sure it will stay that way, but it is quite interesting that we're seeing this with just our long read data. Our lab has done a ton of short read kidney stuff, and we've never seen this, so it's quite interesting. One thing I did do was ask, what is the difference between shared cell types that are split? And across the board for each one, they seem to have changes in alternative splicing processes. So this is really interesting for us, wasn't really expected. And even our gene SRSF7 pops up in here. So that's also really interesting, considering these are just normal kidneys. So we also did transcript level clustering. Again, the advantage of doing this is not only are we getting gene per cell, but also the isoforms per cell. So I just took the cells we had already annotated and clustered based on the transcripts, and we identified 21 clusters and ended up projecting back the gene level annotations or cell types that we identified. And as you can see, each gene cluster is kind of spread across different transcripts and kind of vice versa. One transcript cluster actually identifies different gene cell types. And again, I haven't delved too much into this, but this is a really interesting area for us to kind of start to define the transcript level landscape of kidney. We also wanted to compare our long read data set to a published short read data set that was also using single cell. So we used this paper here. Again, it's a mouse kidney, and we just did this confusion matrix to look at consistency between what we called a cell type versus what they called it, and pretty decent consistency across annotated cell types. And I will say, with the new implementation of Dorado, even from that part of the analysis has greatly improved this based on, I don't have the other data set, but it looks a lot better. So I think everything seems to be improving for this kind of workflow. And then finally, the entire point of this is to look at alternative splicing between cell types. So for the sake of brevity, I'm just going to focus on looking between PCT1 and TAL1. And just to remind you guys, these have different spatial locations within the kidney, and they also have different functions. So to do this DTU analysis, we use this package called dturtle, which is specifically made for single cell analysis. It isn't necessarily for long reads, but we did use it for that. And this package identified almost 600 genes with differential transcript usage among over 1,100 transcripts. And these are the top 10 genes that, most significant genes that were identified between the PCT and TAL. And so we're going to focus on Claudin 10. When we saw this, we were really interested because Claudin 10 is actually a very common TAL marker, and we see it come up for PCT as being significantly differently used. And so from this package, it gives you this kind of output, which kind of gives you the structure of the isoforms that are changing and tells you to what proportion. So in this case, the 201 transcript is used 90% more in the PCT versus the TAL, whereas the 203 is used 90% less in the PCT, or it's used more in the TAL. And so to kind of validate this finding, I designed probes for in situ hybridization and performed that in mouse kidney. we can see claudin 10 didn't come out very well, but we would expect it to be in the cortex because that is what this is telling us, that it's in PCT cells. And then claudin 10b we do see is very clearly in the medulla, so we're able to, especially when genes have different spatial locations, can validate this with in situ hybridization. And finally, this finding is not new. Actually, in 2009, it was published using different methods that the claudin 10 isoforms are spatially located in different spots. So using RT-PCR, they had showed claudin 10, again, is in the PCT, and claudin 10b is in the medullary thick ascending limb. And when I look across our data set at the two different transcripts, we can very clearly see the separation of claudin 10A in just the proximal tubule cells and claudin 10B in the TAL cells. And the reason this is important is it leads to a functional consequence of the different expression of these isoforms, and that is when it comes to perm selectivity or preferring anions or cations. So in the case of claudin 10A, it has this extracellular loop domain, which has a different sequence than claudin 10B. And so in 10A, this loop sequence has a net charge of +5, so it will prefer anions. And then claudin 10b has this net charge of -1, so it prefers cations. And because of this, the cells now have different functions along the nephron. And so just really highlighting the importance of doing this. And of course, we want to find novel isoform switches that could have functional consequences that we can test and validate in vivo or in vitro. So with that, I'd just like to summarize and lead into the future direction. So I've shown that loss of this factor, SRSF7, in proximal tubule cells upregulated inflammatory pathways and decreased cell proliferation. More exon-exon junctions were detected with bulk nanopore sequencing samples, and this allowed us to discover differential transcript usage that isn't found with short read. Our single-cell nanopore sequencing showed these split cell type clusters that we're still trying to understand and define. And finally, DTU occurs between the PT and TAL in mouse kidneys, and they do have functional consequences. And the future directions of this work is to investigate alternative splicing in mice that lack SRSF7 in the kidney, because this would help support our hypothesis that this will cause renal dysfunction, and supporting that changes in alternative splicing can drive kidney disease. And so with that, I'd just like to acknowledge the Humphreys Lab at WashU. Also, a special shout out to Jennifer at Nanopore, who helped me with the base calling, and then my funding sources. Thank you.